Now recording. Our lesson today is going to be talking about booklet pages 16 and 17. This is just relevant to your green booklet, but it might come into play when we look at your upcoming assessment. Now, the key things for you to note, DNA replication is not a huge part of your textbook, but it is an important part nonetheless. So tonight, I would really suggest going to page 233 and having a read. And you'll note that the question, and I do say question that I've put aside for you, is not too extensive. However, there are multiple questions at the back of your green booklet on this topic, so please go and have a look at them anyway. Skipping over our quiz, our keywords for today, and I'm going to highlight one in particular, but before we get there, we have DNA helicase. We've heard of helicase before. We now have DNA polymerase. DNA polymerase is different to what we've previously seen. We've previously seen RNA polymerase. So today we will look at DNA polymerase and how it is different to RNA polymerase. And our last key term for today is a new one. We haven't seen this enzyme before or this protein before. It is DNA ligase. We'll explore each of these throughout the lesson. Not a long one today. To start us off though, let's define DNA replication. When we replicate something, it means to copy something. Thus, DNA replication is simply where we produce an identical copy of DNA. The key parts here, replication meaning to copy, but the essential part here for us, something that's really important for us as a species and as a living organism, is that it produces this identical copy. If our DNA replicates and it is not identical, it can lead to a whole host of nasty issues, and we'll talk about them later on. It could also mean that it's completely null and void and that it actually makes no difference whatsoever, but we will explore those. The purpose now of this replication, of this copy, is before our cell divides in our next booklet, we need to make sure that each copy of our new cell has the same genetic information as it did previously. So we're kind of jumping the gun a little bit here, talking about cell division when our cells reproduce in a process called mitosis and meiosis, which is hopefully a reminder from year 10. And this ensures that both cells receive a complete set of genes or a complete set of DNA. So one set of cells which are constantly replicating would include epidermal cells, things that line your internal cavities or, for instance, your skin. The reason that we are constantly having DNA replication occurring is because we're constantly producing new cells. Why are we constantly producing new cells with our skin? Because it is constantly being worn away. Here are our three key enzymes for today. I mentioned them on our post-it note. These are the really essential ones that I need you to know. So let me zoom in right here. Number one, helicase. We spoke about helicase the other day when we looked at the process of transcription. Helicase, we mentioned, breaks the hydrogen bonds held between the two DNA strands, or more specifically, between our complementary base pairs. When we look at this, we know previously that adenine binds with thymine and that cytosine binds with guanine. Helicase is responsible for breaking these bonds here. Put a little X in there. Breaking those bonds, breaking those hydrogen bonds. A common analogy for helicase, therefore, is to think of helicase as a pair of scissors. Or I like to think of it as a helicopter, because what would happen if you went and stood up under a helicopter as it was rotating? It would chop you in half. So ideally, don't go stand up next to a helicopter, but also when we're talking about helicase, it is, think of it like a pair of scissors. Our next one, and I'm going to highlight the start of our second enzyme, because this is how it differs. We are now talking about DNA polymerase. Why are we talking about DNA polymerase? Because it is what adds our new or complementary nucleotides to our DNA. The last time we spoke about polymerase was RNA polymerase, because it is adding nucleotides to mRNA. So the type of polymerase that we have just tells us what it is building. And in this case, DNA polymerase is going to be really important for helping us to build a new piece of DNA. Our next one, and our final enzyme for the day, is ligase. If helicase is like our scissors, ligase is like our glue stick. Ligase glues the DNA back together. It reconnects, or connects up anyway, our sugar phosphate backbone. It helps to rejoin our DNA. So helicase comes through and it breaks the bonds. RNA polymerase brings along and helps us to form a new strand of DNA, and DNA ligase stitches everything back up together. What we'll look at now is a nice image, and I'd really strongly suggest that you have two colours. So ideally 2 highlighters or 2 pen colours. Here's the diagram from your book. What I'm going to do is I'm going to draw my original strands, and I'm just going to follow the sugar phosphate backbone all the way through to the end. So if I start on the left-hand side here and I just follow this particular strand, and I follow it into this spot where it is replicating, you'll see that one strand of our DNA continues all the way into our new strand. Our second strand, if I highlight it in the same colour, I know it's a little bit messy on the left-hand side, but now we can see up the top, it's now its own distinct strand as well. These are our parent strands. They are from our original molecule of DNA. Within these strands, I'm not going to highlight them, but maybe I'll just introduce a little bit of colour. We have all of our nucleotide bases. So maybe if I just draw some in here, and we can see that our nucleotide bases pair up with one another. That isn't going to change. What I will add now, though, is in a different colour. Maybe we'll just use yellow for the sake of ease. Our yellow is going to follow our new strand. This is the strand that we are creating. And we can see I will start with the bottom. Our bottom strand here continues all the way through. Apologies for the overlap. This is one of our new strands. Our second new strand is this one in the top row, again in yellow. Notice that when we create a new set of DNA, we're not going from one DNA into 2 separate brand new pieces of DNA. We simply divide our DNA in half, and we use the information that we have contained in half of the DNA to complete our set. So this is a very simple diagram, and what we'll do now is we'll move on to the next diagram in your booklet, and we'll fill in and we will label some of the key components here. So what we're going to do is we're going to start up in the top left-hand corner. We have some free-floating nucleotides. So we're just going to call these our base pairs or our free nucleotides. Why are they free? Because they're not bound to anything. They are floating around in our intracellular solution. Our next part, I'm just going to circle it briefly, is this enzyme here. It almost looks like it is cutting our DNA in half. So which protein or which enzyme do we think this will be? Helicase, fantastic. This is our helicase. From here we have one other protein, and it is our protein in yellow. It is in 2 spots around our cell. 1 and 2. What, therefore, will this one be? We're going to label it up the top right-hand corner. It will be DNA polymerase. I'm going to write DNA and then I'm going to highlight it just to make it really clear that this version of polymerase is the one involved in our DNA. From here, we're now highlighting 2 different strands within our DNA molecule, or within our 2 new DNA molecules. Number 1, if we follow, and I'll use my red pen here to follow this piece of DNA all the way through, we see that this one here is our original strand, which means that this strand that we're trying to label up the top of our diagram is actually a new strand of DNA. Okay. So this is our strand which we have produced. It is our new strand. So we'll just write in here simply, this is our new strand. We'll introduce some terms to describe this a little bit later on. And it also means that if we do the same pattern again and we follow through from the top, follow through our diagram, that this is going to be our original or our parent strand. Okay, so we're just going to call it the original strand for now. Now that we've labelled our diagrams, now that we can see roughly what's happening here, let's break it down step by step. I know you've got some space in your booklet to summarise these steps involved in DNA replication. And it is just going to be these 5. That is it. So starting at the top for us, we've been introduced to helicase before. We know our helicase unwinds our double helix. But it doesn't just unwind, it also unzips. It helps us to both unwind and unzip that double helix. What this results in for us is separation between our 2 strands of DNA, thanks to the helicase's ability to break those hydrogen bonds. And those hydrogen bonds are occurring between our 4 bases. Once again, I'll write them off on the side. Adenine and thymine, guanine and cytosine. Now that we've unwound our DNA, all of our bases are exposed. And this is great news for our DNA polymerase. Each strand, so both our template and our coding strand from that original parent molecule, are now going to act as a template. So both of these strands will act as a template. This then leads us into our second protein, which is DNA polymerase. And DNA polymerase, same as RNA polymerase, is what comes along and adds all of those complementary nucleotides, and it matches them up based on these pairings that we've done up the left-hand corner or the right-hand side of our page. So again, adenine and thymine like to bind together, and we also have guanine and cytosine which like to bind together. Our 4th step here, to help us to re-zip our molecule, hydrogen bonds begin to reform, or form for the first time I should say, between our complementary bases, and that is on our original and parent and our new strands of DNA. So we end up with 2 strands or 2 different pieces of DNA now, one from the original or from the parent, and one which is a brand new molecule. Lastly, DNA ligase rocks up. DNA ligase, as a reminder, is like our glue stick, and it helps to connect that sugar phosphate backbone of our newly synthesized strands, and everything is all finished.